I'm going to talk basically about, I haven't got time to cover anything more than the urgent issues, within season issues that have come out of the research that we're doing at Curtin. Basically, I'm talking about work that's been done by Caroline Moffat's group, or Simon Elba's group, and Fran Lopez's group, who are within my ACNFP group at Curtin now. So I'm basically going to talk about 4 areas which have come up during the course of this last growing season and are developing in interesting ways. The first one is, has mace resistance to tanspot or yellow spot broken down? I'll then talk about barley powdery mildew from 2 perspectives, which varieties we recommend on the basis of the pathotypes of the disease, and which fungicides are recommended on the basis of fungicide resistance. And I'll say a little bit about the new fungicides that are on the way that we so desperately need. So the first question is, has mace resistance to tanspot broken down? We persist in calling tanspot, like everyone else in the rest of the world. In Australia, it's sometimes known as yellow spot or yellow leaf spot. The disease is caused by a Pyrenophortridis aeropentis. Now, this is the most important disease of anything in Australia. Interesting, I heard the talk before lunch, just confirming the numbers that were widely disputed, that this is a really big disease, causing averages of 300 kilograms per hectare losses. So it's a really important disease, and it's been important that we can make sure that we have suitable resistance in the germplasm. And we've been working with this disease for about the last 4 or 5 years now, and what we have been able to do is to rationalise how this disease works. Now, if you went to university and studied plant pathology, you would have heard about the gene for gene hypothesis, and this is this diagrammed in the top here. So here in the case of the biotrophic diseases, so the rusts and the mildews, you have a system where the fungus is detected by the plant if it produces a particular molecule, and that detection leads to resistance. So if the fungus loses that molecule, or the plant doesn't have that receptor, then you get disease. So disease essentially is the negative situation, the absence of recognition. The necrotrophic diseases, tanspot and staganospora and net blotches would be examples. We have the opposite situation where the fungus produces a molecule that's recognized by the host, and that causes disease. In the absence of the recognizer, or the absence of the molecule produced by the fungus, you get at least less disease, actually, rather than just no disease. So basically, biotrophs are liable to, resistance to biotrophs is liable to break down. Resistance to necrotrophs, such as it can be accumulated, is not liable to break down according to this model. So we were surprised when we started hearing reports that mace resistance in mace to tanspot was breaking down. So mace, this is all the barley varieties, all the wheat varieties, and mace is right up here, quite close to the top. It's in the MR/MS category according to the DEFRA ratings. And that's consistent with being insensitive to this major protein toxo, which we've been working on for a few years now. A very clear case here where effector sensitivity is strongly correlated with disease resistance. So we were surprised when we started hearing about mace breaking down. So what we saw was we were getting reports of tanspot-like symptoms on mace, and the question was, is this really yellow spot? And I think, as been alluded to in Andrew's talk, it's actually quite difficult to reliably differentiate these diseases in the field. So what we did is we collected these leaves, went down to Esperance and got them back. And in fact, the majority of fungi we isolated from these lesions were not tanspot at all; they were alternaria. And so we did occasionally see tanspot, but mostly with other pathogens as well. So the question is, what proportion of the symptoms we are seeing are really due to tanspot and really due to this other pathogen? And just to show you that this other pathogen does actually cause disease on Yip, which is a very susceptible variety as far as tanspot is concerned, but also in almost the same extent on magenta, which is resistant to tanspot. So when we're now at the stage of doing Cox postulates on this pathogen, it may be that we have basically a new disease, or we may have a disease complex, which is giving us disease on mace, which previously was doing so less. So in conclusion to that little section, we're still gathering information. We may have a new disease, and for those of you who are out there, if you see tanspot-like symptoms on varieties that are in the MR or MRMS category, then we'd certainly be very interested to receive those isolates and study them further. Okay, the second thing is this barley powdery mildew sort of things. Now, barley powdery mildew is a lovely disease, and it can cause very serious amounts of disease. And we've had over the last, well, I don't know what, 4, 5, 6 years, what can only really be described as an epidemic. We estimate about $100 million a year has been lost in 2010-2011 seasons. So that makes it one of the top diseases of anything in Australia. So why have we had a barley powdery mildew epidemic? The short answer is we've had a lot of varieties that are very susceptible, and we've had a single fungicide class being used to control the disease when it does occur. All the fungicides have triazoles in it, a couple have a mixture with triazole and strobilurin. So how can we actually reverse this epidemic? It should actually be quite a straightforward thing to do, and I think with GRDC's help, we're putting things in place both from the genetic perspective and the chemical perspective to put the disease to bed. So we have, the strategy really is to introduce or support the introduction of new resistant barley cultivars, and that's happening, and we're supporting that, to register new fungicides, and we're helping with that, and also to manage the existing fungicides, and all of these things are going to be part of the solution. So first of all, just thinking about the genetic side of things, we've got a number of different ways we could go ahead. We've got what's called major R genes, but these are susceptible to the boom and bust cycle. We have another major resistance gene, or let's say it's a very effective broad spectrum disease resistance gene, but it has the reputation at least of a yield penalty, although I'm glad to say that all the major companies are introducing, or a number of major breeding companies are introducing varieties with MLO in them. What we've been advocating is because to get around the major R genes problem of the boom and bust cycle, one way you could do it, but we won't really recommend this, is to have multiple major R genes. So therefore the fungus would need to lose multiple avirulence genes. So that requires that you know what avirulence genes are present in the population at the moment. In the longer term, we advocate going for minor R genes and large numbers of minor R genes, and maybe, but both these strategies require that you know what pathotypes are present in the population. So as I put up this diagram here, what we need to know is which avirulence genes are being produced by the pathogen. So what we've done is we've collected isolates from all around the state, and we did this by driving out there, but also by involving lots of high schools, which has been very good fun. And then what we can do is ask what percentage of these isolates that we've collected cause disease on barley lines carrying known resistance genes. And what you can see is there's hardly any resistance genes which are completely effective. One here is MLO, we've talked about, and there's one or two around which are at the 0 level. Quite a few are at relatively low levels, so less than 30% of isolates are present, but there's lots of resistance genes that have got heading towards 100% of the isolates can cause disease on those resistance genes. So what we've done is we've looked at the current list of barley cultivars, tried to work out what resistance genes are present, and then come up with a series of recommendations. So if the cultivar itself is commonly infected, we've not recommended it for barley mildew prone areas. If the cultivar is either occasionally infected, or all the individual resistance genes we know are in it are individually broken down, defeated by pathotypes that are present, then we don't recommend it for mildew-prone areas. At a slightly less stringent recommendation, we say you can grow it on your own risk, as it were, but you'll probably need to protect it with suitable fungicides. If we know that the cultivar relies on 2 or more effective major resistance genes, we're basically saying it's a temporary recommendation, but vigilance is required because either of those resistance genes could break down quite quickly. If the cultivar has multiple minor R genes, we would recommend that, but we don't know about any of those. Or if the cultivar has MLO, we would recommend that. And this, I would just say, is just from the perspective of barley mildew. I know growers need to choose their varieties for a whole variety of other things, but we're just focusing on barley mildew. So on that basis, we've come up with this traffic light concept, and we've just mentioned Bodan down here, right at the bottom of our list. It has a defeated resistance gene, A8. Hindmarsh has just been mentioned. It's rated as MS, but its virulent pathotypes are widespread. Up at the top we have the MLO-containing varieties, which should be no problem from our perspective. We have a couple of varieties where we're not exactly sure what resistance genes there are, but our recommendation is that is okay, at least for this year, but we'll need to watch it on a season-by-season basis. And then we have a couple of varieties, Bull Oak and Jaegen, where the individual resistance genes have broken down in the case of Bull Oak, or let's say cultivar isolates that can infect LA or A7 are quite widely found. And then Jaegen, which has broken down, as I observed in several areas as well as driving around the wheat belt. So that's our recommendation, and of course these talks are available and may even go out as a press release, who knows. Finally, the third thing to talk about is the fungicide system, the fungicide situation. So fungicide resistance has been detected overseas for essentially all the major diseases that we deal with in Australia, grains, except for the rusts. In the case of barley, in the case of Australia, we've detected barley fungicide resistance in both barley powdery mildew and in Botrytis cinerea. The reason is we've got this extraordinarily restricted range of fungicides, and you've seen this list before. All of these fungicides that are registered for cereals contain a triazole or a mixture of a triazole and a strobilurin. We've heard earlier about spiroxamine or Prosper that was registered for one season, and we are hoping that will be registered again for the following season. So fungicide resistance can occur through various mutations, deletions, insertions, or alternative pathways, and decreased accumulation of the fungicide and or expression of the fungicide. Basically what that means, it boils down to, is that triazole fungicides, you can detect mutations in what's called the CYP51 gene. These are when you have these mutations, they're characterized by low resistance factors, i.e. the mutant strain is inhibited by maybe 10 times the amount of fungicide as the wild type strain is. We would call that tenfold a low resistance factor. You can accumulate multiple mutations, and as you accumulate mutations, the resistance factors increase and the range of fungicides that are infected also increases. So our nightmare scenario is a kind of staircase to hell where the selection by the fungicides selects for mutations that gradually erode all of the triazole fungicide effectiveness. The QOIs are strobilurins. There's a single mutation commonly found and it gives a large resistance factor. So this means that the triazoles, you tend to see a drop-off in your performance rather than a catastrophic fall-off. In QOIs or strobilurins, you tend to see a catastrophic drop-off. And that's what we've seen in the grape powdery mildew world. So what we've been doing is collecting powdery mildew isolates since 2009 and measured what we call the EC50, which is the effective concentration or the resistance level. And we've also sequenced the target gene. It's a bit of a complex slide. But what we have here is a series of isolates of barley powdery mildew tested for their EC50, so their resistance level to tebuconazole. And what you can see is that there are some isolates that are right down here at the sort of few micrograms per mil area, and there's other isolates up here at the 20-30 micrograms per mil. So this is what we mean by resistance factor of about 10 or so. And what you can see is the colour. Well, this is another just way of graphing that result. So you have some isolates down here and you have some isolates up here. When we sequence the genes, we see 4 different gene sequences. But what seems to be important are 2 mutations. One is this one called Y136F. Every single isolate we isolated in Australia, which is several hundred now, has got this mutation. This is a mutation that was described in Europe as causing strong resistance to triazole fungicides 20 years ago. Everything in the whole of Australia has got this fungicide-resistant mutation in it. But that wasn't a very significant, or in field terms, not a very significant fungicide. What a mutation. What appears to be significant is this one called 509T. See, the ones that have got 509T are all up here. The ones without it are down here. So the 509T is a mutation that's occurred. These isolates are from Tamworth, Tasmania, Wagga, and Esperance. All the rest are from other parts of Western Australia. The question was, if you look at these two genotypes, the ones with 509T and ones without it, so here we have 4 isolates in the so-called star graph. So here we have all 4 isolates have the same fungicide resistance to azoxystrobin, the same concentration of azoxystrobin, so strobilurin. So this mutation doesn't affect this strobilurin resistance. Now each one of these spider's webs is a doubling of the EC50, and you can see that the isolates with the wild type at 509 have a 246-fold lower resistance, so 248, I should say, fold lower resistance to tebuconazole. But every single one of these triazoles is actually affected to a smaller or lesser extent. So this mutation has affected the resistance to all the triazoles, but tebuconazole is the most important, and the most important mutation appears to be this 509 position. So this was a new result not found anywhere else in the world. And what we did in 2009, we had only a small number of isolates, but they were all the wild type. The following year, only 2 were the wild type and all the rest were the mutant. And this year, actually the ratio is about the same, but we've got a smattering of the wild type and all the rest are the mutant forms. Or to put it another way, we've gone from one genotype 1 year to the following year, almost a complete replacement of the different genotype. The danger, this is what I keep trying to emphasize, is that the danger is that further mutations will compromise the other fungicides as well. So in terms of fungicide in relation to these cultivars, what we're saying is the cultivars up here don't need a fungicide. The cultivars in these 2 areas need to have an effective fungicide for vigilance reasons. The cultivars from this area will definitely need a fungicide if you choose to grow them in a mildew-prone area. So which fungicides to choose? These are the fungicides that are currently going to be registered for this year unless we get spiroxamine as well. As we say, all of these fungicides are affected by the 509 mutation. The field efficacy depends on the resistance factor and what you might call the safety margin. So many fungicides are sold at 10, 20 or 30 times more concentrated than they're required to kill barley powdery mildew. The rate they're sold at depends on which is the most difficult to kill fungal disease. So it could be that there's a safety margin of 10, 20 or 30-fold. So whether the fungicide is working or not depends on the resistance factor caused by that mutation and the safety margin that was built in the first place. It's clear that the worst effective fungicides are tebuconazole, flutriafol and triadimenol, and the best current ones are fluquinazole, epoxyconazole, prothia, propiconazole and ciproconazole. Now you notice this doesn't correspond to old versus new triazoles. So propiconazole, an old triazole, is actually one of the most robust ones still going around. The QOIs so far are unaffected. So what we're saying for fungicide recommendations is avoid products, the solo tebuconazole, flutriafol, triadimenol products. Use full-dose seed treatments based on fluquinconazole, which seems to be still working. Use products, actually my next recommendation is don't use a triazole, but I can't really get away with that because you really haven't got a choice. But if you must use a fungicide, use fungicides with propiconazole, epoxyconazole, prothiaconazole, ciproconazole or QOIs. Use mixtures and alternations, so mixed products, alternations, don't use the same fungicide twice. And use the minimum effective dose of that fungicide. So don't, just because tebuconazole works if you put 3 times as much, please don't do that, or at least we're not recommending that you do that. Use a fungicide that works, like propiconazole, and use it at a dose that is sufficient to kill the disease, but not anymore. And then use non-triazole products if available. Which brings me to my 4th point. We've recently started what we call the New Active Project. This is a collaboration with FAR and Calyx and all the various chemical companies to seek out new fungicides for registration in Australia. It involves new fungicides for cereals and pulses. We're rather constrained by confidentiality because we're dealing with all the wonderful companies. I don't know how many of them are in the room. But I can just give you a peek into the future. So this is the disease level, and these are yields. This is untreated control. This is tilt or propiconazole, pretty effective. But you can get about another 1/4 of a tonne with I had to call it product A and product B. And wild horses won't drag from me what those products are, but they are not far away, I would like to think, from being registered. So, in conclusion, fungicide resistance is here to stay. We need to manage our existing and introduce new fungicides. It's not the end of the world, this barley powdery mildew issue. It can be quite easily controlled. We need to use our IPM principles, use resistant cultivars as your first option, maintain crop hygiene as you always do, and use fungicides in a responsible way according to the label and according to the recommendations. But keep vigilant for new diseases. Cultivars or fungicides can break down. Diseases can spread in from weeds, or they can come in from overseas. And if you put all these things together, we should be able to keep disease levels at an appropriate and manageable level. So I'd just like to acknowledge particularly Caroline, Simon, Fran and Madeline, who did the work in my lab, people from FAR and Calyx, Grey Platts and Kith supplied us with isolates, and then the various other people who were involved in the work we've been doing. Thanks very much.
DAFWA has been offering over the last dozen or more years disease ID training for agronomists on an annual basis. Is there a responsibility for agronomists to only provide advice in things they are skilled in, or does it fall to farmers to ensure that the people they employ to give them advice demonstrate their competence in that?
I could tell you a story about Staganospora in the UK. When I first started in plant pathology, Roland would know the story as well, that there used to be one disease called Septoria, and it divided into Septoria tucicide and Septoria nodorum, and people actually couldn't distinguish them apart until the molecular biologists got to grips with it. I would argue that it's hard to distinguish Septoria tucicide, Paranophyllus tucipentis, and Staganospora nodorum, particularly if they're mixed. And what we actually need, if we're going to get to grips with this, is quantitative molecular tools to identify, differentiate, and quantify those diseases.
So is a solution some kind of rapid test that we can access?
It would take a bit of research. Well, the research is not desperately difficult, but what would be difficult would be to turn it into a service that, a diagnostic service that is meaningful in the context of the time it would require to get a diagnosis. I mean, at the moment, it essentially doesn't matter which disease you've got. Your response to it as a grower is going to be very much the same.
Spray it tomorrow.
Spray it tomorrow, yeah. Or spray it at the right time.
Yeah.
I guess it's not really necessarily a question, but is there a way of this physiological versus fungicidal, a quick test where you can just say it is a fungus? At the end of the day, it doesn't matter what it is, it requires a spray, whereas physiological, we can say no, there's no fungal activity here, and say don't spray. I mean, is there something we could do like that? It's not overcomplicated at what disease, we still have to spray it. So.
A PCR test realistically should be done within 24 hours. Particularly if you just want to know it's something, as opposed to exactly what it is. But it would take a bit of resourcing. Maybe the fungicide companies could help resourcing.
Dave Extenia, I'm an agronomist from New South Wales. We're just facing herbicide resistance at the moment, and the message that comes through there is if something is good, don't stick to it. Now, in canola, we're just using butyriaphol jockey, butyriaphol jockey, butyriaphol jockey. In wheat with stripe rust, we're just using tebuconazole, propiconazole, temaconazole, propiconazole, maybe a bit of opus. Aren't we heading for disaster by not having a rotational system? Is it serious? Should we be really considering? Because no researchers, no one's coming to agronomists and saying, look, you're heading for trouble there. We're following one of the golden rules we shouldn't follow, which is we're just using the same thing over and over because it's working well and it's doing a good job. Do we have options, or do we just continue until it crashes?
I'll pick that one up. I mean, I agree. Basically, I think, just to say something on the other side of it, blackleg Leptospira macans is rated as a low risk of fungicide resistance. and the triazoles are medium risk, so you kind of low times medium. But nonetheless, you may be sort of slowly driving towards the chasm rather than quickly driving towards the chasm, which you do with the powdery mildews and the strobilurids. So I don't think we're going to fall over, or my guess is we won't have a massive problem very quickly, but why not have a problem at all, and let's introduce another mode of action into the fungicide for canola. And I would argue that one of the SDHIs would be sensible, but we have a regulatory authority in this country which is very reluctant or makes it very difficult to register a new fungicide, and therefore the market has to be there in order to justify those regulatory costs. So it's a bit of an issue, but I would say that the group who are working on that, which we have a small part in it, we're doing the monitoring of the CYP51 gene sequences in the blackleg isolates, and we haven't seen any changes yet. So we can keep an eye on it, but I would argue that the best thing to do is genetic resistance. The second best thing to do is registration of alternate modes of action of fungicides.